Hey Mustangs, in this video we're going to be taking a look at protein structure. Now we've talked a lot about structure and how that affects function, and that's really important when you're talking about molecules and in all the macromolecules as well. And with proteins it's no exception. So proteins, if you remember, do some of the most functions in the entire body. So with proteins, structure is super important. It's very sensitive, in fact. One small change in the connection of monomers in a protein can change the overall shape and therefore the function of the protein. So even small, minute changes can have huge, massive effects on a protein. So protein structure is very important. If it's not the right shape, it cannot function. It will not be able to do its job. And remember, there's multiple jobs that proteins do, and that applies for pretty much every single protein that we've discussed. So let's go ahead and take a look. First, a quick reminder of an amino acid. You have to understand amino acid structure in order to understand the overall protein structure. So here, if we take a look, if you remember, we build our amino acid. You have your amino group, your carboxyl group, and then your R group. The R is the variable, and that's the one that could change. And between all 20 different amino acids, there's different things connected in that R position for each of the 20 different amino acids. Now, there's different ways to represent these. So here in purple, this would be the base part right here, the backbone basically of the amino acid, and then this would be an example of an R group. Now, in order to connect amino acids together, we do the good old dehydration synthesis. And with dehydration synthesis, remember, that's taking a H group from one, or sorry, a hydrogen from one, an OH group from another, and combining them to make water. And by removing those, you can form a bond between two amino acids. In fact, that's called a peptide bond. So if I connect amino acids together here, so you can see this is a different amino acid in this case because the R group is different. Remember, there's 20 different amino acids, and when I connect them together here, we start to form a polypeptide. So several amino acids connected together builds a polypeptide. One thing to note about the polypeptide is the location here of the backbone. So let me go ahead and mark that. Okay, so you have the backbone here, the purple part. Okay, that's the backbone, and that's going to be very important, that there's going to be interactions between the backbone and parts of the protein molecule. And then the R groups. Okay, so there's an R group right here, R group there, R group there. Okay, so those are going to be very important parts. So you take amino acids, we have 3 amino acids here, you connect them together, and you're building a polypeptide. So there's actually 4 levels to protein structure. The first level is called the primary structure. And the primary structure is actually very simple. It's basically which amino acids are you connecting together in what order. So we can say it's a sequence or order of amino acids that you connect together. So if you take a look at this picture here, there's actually 127 different amino acids put together right here. Okay, so 127 amino acids put together. You can see some of them repeat. So right here, ALA, and you can even find it here. So those amino acids can be used over and over again. The different types of amino acids can be used over and over again. And that overall order that we put those amino acids in is the primary structure. And that's going to be key for the rest of the structure of the protein. So the overall 3D structure that we get for the protein starts here with the primary structure, and exactly which amino acids are we connecting together. Okay? Now, what determines that sequence of amino acids? It comes down to the DNA instructions. So your DNA actually has instructions that say which amino acids to connect together in exactly what order. And that's why your DNA is so important. So when we talk about mutations in DNA, we're usually saying that the DNA instructions change, and as a result of that, it changes the amino acids we put together. Most of the time we find that when that happens and you put together the amino acids together in the wrong way, it causes a protein that doesn't work, and as a result there's usually a problem, some sort of disease in the body. If we are talking about a mutation that is favorable, which is more rare, it could happen, but changing the amino acids could create a better protein, but it's highly unlikely. So that primary structure, again, is simply which amino acids are you connecting together in what order, and that's determined by the DNA. Alright, so an example of the primary structure. So if we took just 7 amino acids out of the 20, so 7 of the 20 amino acids, and we put them together 7 in a row in multiple combinations, you can get 1 billion possible combinations alone. So here put together 7 of the amino acids. Okay? That's an example of amino acid sequence. Those are the exact amino acids put together in a specific order. Changing the amino acid sequence would be this. So I could take those same 7, mix them up. I've changed the order, the primary structure here. I've changed the primary structure. I've changed the order or sequence of amino acids. Okay. As far as diseases goes, this shows how important primary structure is. One single amino acid being wrong in the sequence can lead to disease. So here, this actually shows sickle cell anemia. And with sickle cell anemia, this amino acid here is replaced by a wrong amino acid. So here we have a hydrophobic amino acid, or hydrophilic amino acid, and it winds up getting replaced with a hydrophobic amino acid. Okay? So when you look at these, there's one single difference. If you compare the rest of the sequence, Val, His, LEU, all those, they're the same. It's one single amino acid that's off. And as a result, in the end, when the protein goes to fold, it's going to fold here. That's the normal way of folding versus the sickle cell way of folding. So one single amino acid difference in the primary structure, and you have problems. All right, next comes secondary structure. Secondary structure is going to involve the backbone and hydrogen bonds. So parts of the backbone will actually form hydrogen bonds with each other, and there's 2 basic structures that can result from the hydrogen bonding. The first is helical. So it actually causes parts of the protein to form like a helix, so we call it an alpha helix, something like that, okay? And then there's beta sheets, okay? So beta sheets is where nearby strands wind up interacting with each other, and you get interactions like this, side by side, where you form hydrogen bonds in between here, okay? So you have alpha helices and beta sheets. And if you take a look at this picture of a protein here, and ignore the Shutterstock there, but if you take a look here, you can see that if we fold a protein up, not all parts of the protein are in alpha helices or beta sheets, only certain parts. So you can see this part of the protein here is an alpha helix, and then the orange flat pieces here would represent the beta sheets, and then these blue parts would represent parts of the protein that aren't in alpha helices or in beta sheets. Okay, so they're not having those hydrogen bonding interactions with the backbones. Next we have tertiary structure. So tertiary structure is that overall 3D structure that we get in the end. Now these are caused, or this is caused, the tertiary structure is caused by weak bonds, interestingly. So the important structure that we get in the end is caused by weak bonds, and its interactions with the R groups or the side chains. So those side chains, here's where they come in to be very important. So here, this 3D structure of a protein is represented in a ribbon model. So that's a ribbon model we see there. And then here's the same protein in a space-filling model. So putting in all the atoms and seeing how it all fills up in a 3-dimensional space. The ribbon model shows us the beta sheets and the alpha helices, whereas the space-filling model shows us the overall 3D results of all the interactions we're going to see right now. So the major interactions that cause that final folding of a peptide chain into a 3D structure are hydrogen bonds. So again, these are all weak ones. These aren't covalent bonds. These are weak bonds. So we have hydrogen bonds, disulfide bridges, hydrophobic interactions. So all the hydrophobic side chains are going to want to be towards the middle of the protein. So that's something that's really, really important right there. All hydrophobic side chains want to be towards the middle. They don't want to be exposed on the outside of the protein. They want to be towards the middle with other hydrophobic side chains. Then there's van der Waals interactions, and then ionic bonds. So those are the major interactions that will happen that will fold the protein, the polypeptide chain finally, into a 3D structure. Okay. Actually, let me go back real quick. So let me use my example here. So let's say you have a primary structure, a certain sequence of amino acids. It's going to start folding into alpha helices, beta sheets, stuff like that. And you might wind up with an overall structure of the protein that looks, something like this. Okay, so you have your tertiary structure of the protein, and the reason why it's like this is because all the hydrophobic side chains will go towards the middle. You'll have hydrogen bonding going on between side chains, ionic bonds, disulfide bridges, and you get a structure like this. Now, here's where primary structure affected tertiary structure. If I had changed just one single amino acid, that would change the overall structure. So now maybe I change it and have something like this. If I change a couple more, now instead of the structure being like this, I might have something like this. Okay, so the amino acid sequence is going to affect how this protein is going to fold in the end to get its tertiary structure. So most of them have some sort of globular structure going on, three-dimensional structure like this. All right, so finally you have quaternary structure. This comes when one polypeptide chain is going to interact with another polypeptide chain and come together to form the overall protein in the end. Most proteins are made from more than just one polypeptide chain. So this would be a whole bunch of amino acids together in a polypeptide chain. This would be another set of amino acids together in another polypeptide chain, and then they wind up interacting together, and hydrophobic interactions are going to cause them to set up in a very specific way and form your overall protein in the end. So that's usually how most proteins are formed. So most proteins will be either one polypeptide chain or more than one, up to 4 polypeptide chains usually, in order to create the overall structure of the protein. All right, so here's one example here. You have collagen. This is the protein that keeps our skin nice and tight, and it's the one that we start to produce less and less of as we get older. And as you can see, it's actually made of 3 different polypeptide chains working together to form a fiber. We call these fibrous proteins. And then this one represents here a globular protein, and this is hemoglobin. This is the protein that carries oxygen in your blood. All right, so a quick review. You have the primary structure, which is based on your amino acid sequence. So peptide bonds form between amino acids, and it's the specific order of amino acids you connect together. And remember, that's determined by your DNA. Then you have your secondary structure, which is your interactions between the backbone and forming hydrogen bonds, so alpha helices and beta sheets. And you can see here in this picture where they fill in, so the folded parts would be the beta sheets, the loops here would represent the alpha helices, and everywhere else are parts of the protein that aren't folding into those structures. You have your tertiary structure, which is based on the R groups and hydrophobic interactions, disulfide bridges, hydrogen bonds, ionic bonds, and van der Waals. So all of those forces, all of those types of bonds coming together there. And finally, we have the quaternary structure. So quaternary structure is going to involve, remember, multiple polypeptide chains coming together to form the overall structure of the final protein there. Some proteins can be one single polypeptide chain, so that is a possibility, but a lot of proteins are going to be made from multiple polypeptide chains coming together to form the overall structure. And that's usually based, remember, on hydrophobic bonds, with the hydrophobic parts wanting to be towards the inside of the protein molecule. All right, chaperonins. Because of the whole hydrophobic thing, in order for a protein to be built right, it has to be in the right environment. So chaperonins are a place within the cell that the proteins can actually fold properly. So proteins are made inside these chaperonin pieces here. Polypeptide parts go in, and there's a hydrophilic environment. So water-based environment on the inside. That water-based environment on the inside of these structures here is going to allow the protein to fold based on hydrophobic bonds, where the hydrophobic parts go to the inside and fold into the correct overall 3D structure. And then when it's made, it gets released. Last thing is protein denaturation. Okay, so protein structure is, remember, based on those bondings, hydrogen bonds, disulfide bridges, all of those weak interactions, and those can be disrupted. So the 3 major things that can affect those types of bonding are going to be temperature, pH, and salinity. So changing those 3 things can actually change the overall shape of proteins, and that could be a bad thing. So that's why, like, if you've heard those stories of someone drinking too much water, they died, that could actually change the overall pH of the body. You know, really simple things like that could have a major effect, because if you change these 3 things, protein will not be the right shape. It'll actually change shape. So that's what denaturation is. It's actually the protein changing shape as a result of change of temperature, pH, and salinity. And overall, that's going to change the secondary and the tertiary structures. And again, wrong structure, wrong shape is going to equal no function of the protein.